Well, we might get this started because we have so much to share. So, as you know, we, or at least like about a third of us here, have spent the last 2 months on the Investigator. And it's been a very bumpy ride back, so I might hold on to this table still while I'm talking. I've had a few very wavy days at home and just getting my balance back. So hopefully I won't fall over in the next half hour or so. We went to the Cook Glacier region, and when we developed our proposal and going to the Cook Ice Shelf region in Eastern Antarctica, we had a very, very long title, Paleobiological and Paleoclimate Reconstruction of the Cook Ice Shelf, a Genomic Approach. But that very quickly turned into a much shorter title, the Cook Ice Ecosystems and Sediments, and became known as the Cookies Voyage. And so when we developed the proposal, we came together with 13 PIs, thinking about the research that we want to do in this region. And so you can see everyone listed here. They're from Australia as well as from overseas. And so this was the first time that I've been a chief scientist on a voyage, which was really exciting. And a really good experience. So as you can imagine, with 13 PIs, the berths on the ship filled up very, very quickly. We had a very multidisciplinary program. And so you can see here on this list who actually came on the voyage in the end. So again, we've got people from all over the world. I've bolded everyone who will be presenting today because we thought we would make this a little bit of a group presentation. And so one by one, we'll come up and talk about our expertise and the samples and the data that we've generated during this voyage. So this is the team. I think that was probably in the second week of the voyage, and we were really excited and got along really well, but we still do. So everything's good. And lots of us are still here this week, and we're talking about collaborations and papers and new projects. So we really came up with a lot of ideas based on what we collected, and had a really good time. So a bit of background. Why did we want to go to the Cook Glacier region? So you can see on that little map there with the white box, indicated the Wilkes Subglacial Basin, and it's a really important region in terms of climate change. We have 3 glaciers that sit right next to each other, the Cook, the Ninnis, and the Mertz, and they're very vulnerable to climate change. So the Cook especially is, based on recent modeling, probably the most vulnerable glacier of all of East Antarctica. If it melts, we're looking at potentially about 1.5 meters of sea level rise, and the whole region, like the Cook, Ninnis, Mertz, they probably amount to about 3 meters. So it's a significant amount of ice that might be melting over the next couple of 100 years, very rapidly. And so there's a lot of interest in this region from various projects all over the world that are starting to investigate this region more closely. So our voyage was quite biologically focused. We wanted to look at the sediments as well as the ocean. How did the ecosystems evolve over the last million or so years? Million years was targeted because that's based on preliminary data that we have from sediment cores from one of Laura's previous voyages. Looking at how far we can drill with the sediment coring equipment into the seafloor, based on what the investigator is able to do. But it wasn't all just about the biology. It was, of course, also looking at the physical oceanography and the geomorphology. We mapped a lot. And so we really wanted to get a good understanding of the region. What does it look like? What did it look like? And what does it look like now? And where's it going to go in the future? So we had a lot of genetics on board, a lot of animal-focused research, but also a lot of physical and geochemical and geoscience research happening. When we planned this voyage, and as of or on the 1st of January 2026, this is basically a list of things that we thought we would do. I would say it was ambitious, but at the same time, it was also quite reasonable because, as you can see throughout the talks, we actually achieved a lot of these things. So we wanted to collect multi-beam data, sub-bottom profile data. We towed the magnetometer. We wanted to collect seawater samples for lots of different analysis, from plankton to genomics, eDNA, and just chemistry. We were aiming for about 30 CTD casts, 5 trace metal rosette deployments. We were thinking 10 multi-cores, 8 custom cores, 5 piston cores, hopefully, 25 deep-towed cameras, 20 sleds for animal collections. We had 5 Argo floats with us that we really wanted to deploy too. And then we had quite an extensive outreach program that was led by Julene. As we were starting the voyage, this is what we had in mind. So we started from Hobart. It was a Hobart to Hobart voyage. And we knew that the sea ice would be quite extensive in the area. So we had quite a extensive contingency plan also, which meant that we had several stations along the way where we wanted to collect multi-cores and trace metal rosette samples. And so we spent a lot of time on that in the first week of the voyage. So actually within the first week, we already had a number of multi-cores and trace metal rosette deployments that we were working on. Our primary study area was the Cook region. This is really where we wanted to get to. The secondary area was more like in the Mertz area, which is nearby. And then as a third option, we thought we would go to the Balleny Islands, which is a little bit shallower too. So you can see this was sort of some figures that we looked at in the planning stage. We had a voyage track that was approximate, but we kept it really flexible because the sea ice always moves around in the area. And then it could have been that we can get in from the east or the west, or, you know, we just really had to decide based on what the area looked like when we got there. So in the end, after we ticked off a lot of things along the transit and starting our work on the slope in the Cook region, we still had a lot of sea ice. So this was probably the end of January, maybe the 20th or something. And you can see we were always having a good look at that area because the ice seemed to be retreating at about 5 nautical miles per day. And so we thought, we'll just wait a little longer and hang around here. That's when we got a lot of the work on the slope done. So we actually got quite a few good records from that Cook and Mertz region of the slope. But you can see that the ice is quite expensive. And then, you know, the master and the crew, they kept our expectations in check sometimes just by comparing how much ice actually has to still disappear before the investigator is able to go into that region, because it's not an ice-rated ship. And that at some point towards the end of January, that was probably an area the size of half of Tasmania. But we kept ourselves busy, so the ice was moving west, sometimes a little bit further north, then a little bit east, depending on what the weather looked like. And so, you know, we would always go back and forth a little bit and tick off different stations that we had planned. But in case anybody was following us along with the ship track, it might have seemed a little bit crazy because, yeah, we would just literally go back and forth, and, you know, depending on what the ice did or where the wind came from to see what we could do. So then we mapped a lot. You can see there are certain areas we were focusing on. So Luca is going to talk a little bit about that area that we mapped to the northeast. Then we went towards the slope in the Cook-Ninnis region. And then in the end, because we couldn't get anywhere onto the shelf, but we really wanted to collect some animals and, you know, do a bit of work in shallower areas of the Wilkes Subglacial Basin, we actually moved a little bit further west to the Dibble Glacier, which was ice-free, and did a lot of work there. I think that's all I wanted to say for the moment, and Luca will talk a little bit about the geophysics.
Yeah, sounds good. Yeah, we got a lot of beautiful geophysical data. We collected multi-beam bathymetry, backscatter, and subbottom, which is mostly acoustic. So for those who are not familiar, there's acoustic waves we send and record the time they take to get to the bottom of the ocean and come back to have an understanding of the depth of the seafloor through the multi-beam. The backscatter gives an understanding of how reflectant it is, the surface, and it's very useful for coring because if it's high reflectance, it means that the surface is harder. So it's potentially, depending on the coring system, not a great site for coring, but that was very useful information. And then the subbottom profile is something similar, but it allows us to see what's below the surface of the bottom of the ocean. So we got, depending on, again, the reflectivity, you can have a range of penetration. Sometimes this is just a few meters or tens of meters, but then we got some beautiful data, especially on the way back on the Tasman rise. We penetrated until 200 meters, and so there's a wide range you can get to. And then the MAGI, the magnetometer, and these records, you can tow it at the back of the ship, and because the rocks at the bottom of the ocean hold a magnetic signature, and we can record the magnetic intensity to understand both the composition, and then after the composition, we can get an understanding of the age of these rocks as well. I just put that figure up there to remind everyone, if you don't know what it is, a mid-ocean ridge, because we crossed it, which is this one here in purple. And this is where new ocean basin forms. So the crust breaks up and two tectonic plates that diverge from each other. This is not a straight line, there's segments. And all these segments are connected by transform faults, which are where like pieces of tectonic plates move parallel to each other in different directions. And the trend is that these transform faults facilitate these segments of the mid-ocean ridge to get together. And this is an example of the body that we collected where we crossed one of these transform faults. And it's basically the tectonic structure where the red there is shallower and the blue is deeper. And the lines here, the fracture zone, the yellow lines are basically transform faults, but not active anymore. And these files, these shape files are based on papers in the literature. And considering that close to Antarctica, the data is quite sparse, it's very useful to get this type of data set because we can see if they actually match with what's modeled or not. Top right is the bathy with backscatter as well. So the darker parts are the parts that are less reflective, so softer. And when the color is stronger, it means that it's more like white, transparent, and it's very high reflective, and it's a harder surface. So these bathy and backscatter were two of the main data we collected, and this is an example of the subbottom profile. And yeah, so this is the seafloor. The acoustic basement is where, because the acoustic waves travel through the subsurface, but then they dissipate, and then when the energy is dissipated, you have no data anymore. So that's the acoustic basement, and yeah. Some subbottom is, like you can see, very sediment well layered, but considering that here we crossed this tectonic structure, what you get is not an amazing penetration, and yeah, the surface. But yeah, this is basically the geophysics, and this was very useful to plan where to target the coring sites and yeah, CTD sites as well, because yeah, looking at this data, you can understand what's the best spot to do operations.
First of all, I just want to thank all of you, and especially Linda, to have joined this trip, because this was really a pleasure, and it's how we should work all the time, all together, because that's how you understand how the environment works. And so, thank you very much, Linda, and all of you. This map is showing some of the results that we've got, especially in the Cook and in East Mertz region. And this map is a combination of this cruise and previous cruise, because this area is very big, and so one single project is not enough. So that's also the beauty of international collaboration, because we really need to join the effort. It's very expensive, you know, it's difficult, and working in a multidisciplinary way is really the recipe to understand how things work. Or just showing, for one of the examples that we've got is coming from the north and approaching the region, because of the sea ice and because of the complexity of the operation that we were facing. One of the main targets was to try to understand how big is this Cook mega slide complex that was kind of discovered a couple of years ago after our cruise. And with legacy data, we saw that in front of the Cook, there is in front of the shelf depression, which is on the shelf, there is a giant submarine landslide that has not been mapped very well before. So we tried to map it to look at how in detail with the multi-beam, if it's a recent landslide or if it's a kind of an old feature. And so the idea was to sample with subbottom and multi-beam to try to find the bad spot where we could actually reach the upper surface, which is the last time then the landslide moved. And so with PC5 and bunch of casting core, we tried to get this green reflector, which is on top of this huge avalanche. And I think we got that, especially the sediments that deposited on top. So we'll be able to date the last time this margin moved, which has implications in terms of geohazard for Australia maybe, or for the Southern Ocean, for Antarctica for sure, and also has implication in terms of climate and paleoclimate and how the ice sheet builds its own margin and how the ice sheet and the ocean interact in this particular corner of Antarctica. Then we moved to a little bit closer to the continental shelf edge, and we mapped this canyon that was only partially mapped before. And there is a paper done by 2024 who actually was looking at this little feature on the side, the pinky sediment drift, which was shaped by warm water, the circumpolar water, which is flowing upslope and entering the continental shelf. So this particular feature is a proxy for water masses, and that was based only on geomorphological observations. We had one sediment core that we collected in 2022, was studied by Fiorenza mainly, by a big group. And it was only 3 meters long. So with Linda's project, we aimed to go deeper. And so we were very successful. So we got 20 meters long sediment core in this sediment drift. And we can extend our record from the MIS-5. We can go much, much backward in time, but you will know something about this later. And I think it's
Yeah, thanks for everybody being here today. And I'll echo what Laura said about Linda leading a fantastic cruise and also all of our shipmates. It was one of the most collegial cruises I've been on. And I think part of that is shown in the decision that we made to go to the Dibble when we weren't able to acquire any data at all for the people who are doing work on the shelf. And, it was something that we all talked about. We were all really happy about making sure everybody walked away from the cruise with really a really nice set of data. So we did decide to go to the Dibble because that was the closest area that had access to the continental shelf without any sea ice or without very much sea ice. But it fit into our overall scientific objectives in some really interesting ways. The Dibble Glacier region is located at the western edge, the very farthest edge of the Wilkes drainage basin. So it's a little bit different. It's a little more remote. It has a little bit thinner ice, and it ends up being a very shallow shelf as compared to the Cook, the Ninnis, and the Mertz. It has a lot of strong similarities to the Sabrina shelf, except that it's a lot shallower. So it provided us with some complements and also some comparisons to what we were already studying. There has been a little bit of prior work which led us to be really interested in following up on that. And the first was some physical oceanographic work, which you can see on the top from Yamazaki et al. 2026. So we just found this paper when we were digging around for information on the Dibble. And the bottom line there is that the loss of the Mertz ice tongue in sort of the 2010 or so had really big implications for the production of dense shelf water on the Dibble. And so we were interested in looking at those interactions. Maybe we could look at those comparisons. And there also had been a previous multi-beam cruise there by Frank Nitsche in 2018, I think. No, 2015. And he had done a little bit of mapping that allowed us to see, oh, in the mid-shelf, there's a pretty nice basin with a thick sequence of Holocene biosiliceous sediment. And we also had some ideas about where the grounding zones were and some of the stuff that had taken place on the shelf with iceberg turbation. So we already knew that there were some reasons that we could be successful there. Some of the things that we wanted to look at were this expanded Holocene section, which we were able to get. We wanted to look at the timing of the last glacial maximum and the last glacial maximum extent and then retreat on this shelf and be able to compare it to regions on either side, in particular the Mertz and the Ninnis. And then we also wanted a hypothesis that would pull us all together, and this is this refugia hypothesis, which asks the question of where do shelf fauna go, benthic fauna go when ice is grounded to the edge of the continental shelf during glacial maxima. And this area has some relatively shallow regions just seaward of the continental shelf on the continental slope. And so we wanted to be able to find those spots, which Lara will talk about, and then sample them to address this refugia hypothesis. Did they all just move a little bit off of the shelf and then come back when the area was ice-free again? So I thought we did a really efficient, pat everybody on the back, well-integrated and comprehensive field program in a very short period of time. So it was pretty great.
We're dancing here. Okay, so this is a kind of spider, perhaps for you, but it's a multi-beam map, sort of multi-beam map. Lots of gaps to fill in the future. And this is the Dibble. So if you look at the slide and you look south toward the south, that's the continental shelf edge. That's the slope, the continental slope with gullies, and this is a bank, which is a shallow bank. It's 700 meters deep, so it's not that deep, but it's away from the continental shelf. So this is what Emmy was speaking about, the refugia, what we call refugia, because maybe when the ice sheet was advancing on the continental shelf up to the edge, and it couldn't go any further, on this bank, maybe some of the animals, Sally's animals, were perhaps escaping and staying until it's warmer and go back to the continental shelf. So we visited this bank and we did many activities here because of this. But in the continental shelf, the first thing we want to do is to go to the mid-shelf basin that Frank actually discovered during his cruise. And we were able to get there and we got almost 3 meters of cast and go. It's a shame we didn't have a piston core. We could go deeper, but we went through that very finely laminated sequence, which is maybe Holocene. We don't know about the age yet, but it seems to be quite recent. So it will tell us a story of this region in a very high expanded resolution. And then we were able to map the outer continental shelf, and we do see all of these with the ridges. And the big one on top is what we call ice grounding zone wedge. It's a asymmetrical wedge that was shaped by the ice sheet when the ice sheet was actually going over that region, and then step backwards. And this is the bank. So that profile is that one. So it goes from east to west over the top. We sampled the top. We did some SLED and deep tow camera. And also we sampled the base of the bank on the other side. So we visited a whole bunch of regions. We had some hints, some clues on all of them. Of course, it's an interesting area. We need to go back to do some more work. We work on the bank with iceberg scours. We found lots of interesting animals there, and Sally will speak about this. And we discovered the beauty of using the SLED for geology, because we collected such a large amount of kilos of clast, all sort of clast, and makes Luca very happy. Especially Luca, who is the specialist on this, and you probably can tell us some more about this.
Yes, as Laura said, during the deployment of Epidentic SLED, we collected a lot of pieces of rocks. They range in size from few centimeters to tens of centimeters. The rocks are very useful because for two main reasons. The first is because this part of Antarctic continent is, by a geological point of view, very poorly known. So we can study these rocks collected and bringed by glacier and iceberg to reconstruct the geological evolution of this sector of Antarctica and the implication in the ice sheet development. And the other useful things to study this rock is because we can have the fingerprint of this glacier and compare the modern record with the old record we collected in the course. So we can see if there are different provenance indicators from the present day or during glacial or interglacial period. And this is the main target for the rocks. And now we can talk about the next slide of the core.
Thank you. During this cruise, we have the opportunity to collect different sediment core, 5 piston core and 14 Castian core. And for all these core, we collected more than 10,000 samples. We have collected discrete sample for the following analysis: the ancient DNA, microfossil, geochemistry, grain size, geochronology, bulk density, radiocarbon, and clast content. Also because here we found a lot of clast, so it could be very useful also here track the provenance of this material. And then on board we do different analysis: the MAXAS, thermal conductivity, and the preliminary biostratigraphy. And this last one allowed to us to put our sediment core, our sediment in the time. And we are so happy especially about one of these sediment core, because we're dating more older than 2 millions. And another one, which is a very expensive sedimentary record for the entire Pleistocene. So we are able in the future to compare the warmer period and the colder, and which is very useful to understand how future climate could be evolved. And also we collect different core on the Dibble Glacier, which is the completely new area. And this is allow us probably to study the Holocene, but also the glacial transition and the Holocene. So we have collected a lot of very beautiful data. And so now I'm speaking about the multicore.
Cool. So yeah, the multicore and TMR team were sort of in one. We attempted 19 multicore deployments and 12 TMRs, which were very much above our scheduled amount that we were allowed. So we had a transect from Tassie down to the Cook region. We did quite a few multicores specifically for the DNA analysis in the Cook-Mertz slope region. And then we had a transect from the slope to shelf for the multicores and TMRs as well. And sort of the reason behind the multi-core TMRs being in the same locations is to look at the sort of flux between the sediments and the water column of certain elements. Cool. So we had a broad range of samples in the multi-core, very similar to the Karsten and piston cores, except a couple extras. And we also sampled a lot for pore waters, which is our centrifuge tubes, whirled them around and extracted the water on top. And then those samples specifically will be used in comparison with the TMRs, where we sampled nutrients, trace metals, and for a bunch of other analyses as well. Yeah. Here's Izzy.
Yeah, so I'm Izzy and I was part of the physical oceanography team. So as Linda already said, we did 37 CTDs and then we also did 5 rapid casts. The depths, I think were really great. We had a great range of about 300 to nearly 4,000. We also did lowered acoustic Doppler current profiler data for each cast, which tells you about the velocity of the water column and directional flow, which is super helpful when you're looking at different water masses as well, and you can see where they're flowing and where they might have come from. These CTDs broke down into a couple of different kind of sections of the Transit South, 21 in the Mertz and Cook regions, and then 8 in front of the Dibble, which I think, considering the Dibble was kind of a on the fly or kind of on the voyage decision, we got quite a nice little data set there, which will be really informative. And also as part of physical oceanography, we did 5 Argo floats, 1 BGC, 2 core, and then 2 deep. So the sampling process, we had a whole range of different people doing lots of different sampling, and kind of to start with, this would just look like collecting water from the Niskins, and how we decide the depths. We'd look at kind of a cast on the way down, and we had some kind of standard depths that would range between 6 and 12 samples, depending on the depth. And if there was anything else interesting, we could just add that in. So, yeah. And this also just shows kind of a little bit of a distribution, as you've already seen the voyage track, but these are where the CTDs are. So you can see the kind of Transit South, where we spent the majority of our time, and then part of in front of the Dibble. And you can also see over here, we kind of have a couple of areas that we go to a little bit more extensively, which was the back and forth again that Linda was chatting about. And these would be sort of targeted locations, which is also what Luca was alluding to with using the geophysical data. So we crossed a canyon and that looked really interesting. So we went over a canyon in front of the Mertz twice. We did a northern and a southern transect. We also identified a location where we'd seen sediment waves and internal water waves in a previous cruise. So we went there and that's where we used the rapid cast to try and get a little bit of a higher spatial resolution as well. And then in front of the Dibble, we targeted the 132 line based on the 2024 Investigator voyage, which is the paper that came out. So that was really exciting for me to kind of have some data to compare against and also sort of inspire where we might target. And I'll just won't go into detail about these, but these are just the 3 different key locations of temperature salinity plots, and you can just see that they're quite distinct. So I'm really excited to look into these and especially compare the Dibble region to 2 years ago, see if anything has changed. And then, yeah, the Argo floats. We had previously kind of decided where these were going to go, but due to the extensive ice, again, we had to think about this whilst we were out there and use the geophysical data as well as CTD data. So we chose places that kind of had bumpy topography and gas, or where we thought as well for the deep solo, that was the furthest south we could get at that time. But due to changing ice conditions, we actually managed to deploy the BGC further south. We went for the second deep between 2 interesting oceanographic regions, hopefully to capture whether it flows with the ACC or the ASC, will hopefully capture something interesting. And then 2 different areas of the Dibble Basin, again, to see if there's any kind of on-shelf circulation and capture as much as we can before it flows out.
So Fiorenza and I spent a ton of time in the underway seawater lab, which people seem to be afraid to come into. They thought there was all sorts of complicated things on the walls, but we spent a lot of time filtering. And we did this for a variety of different reasons. One, it is completely not to interfere with anything else that's going on, so we can take samples and for lots of different people. And on this cruise, we sampled primarily for ourselves, which was with regard to the planktonic assemblages, whether they were diatoms or silicoflagellates, dinoflagellates. We have filtered for all of those on the transit down, across, and then again on the transit back. And this provides us with information where we can understand why specific organisms live where they do, and then apply that information to our paleo environmental records. It also provides a snapshot for where we are today, and as the ocean changes over time, we can compare things from previous cruises to ones that come on in the future. So we just document what we see, a kind of an observing system and a documentation of way the ocean lives today. We also collected samples for microbial communities, for eDNA. We collected samples for hydrochem, which were very kindly analyzed for us by Pavi and Narendra. And then we also collected samples for Neve for her Fragilarepsis cruenlensis shape analysis to try to understand the use of that particular proxy for paleo temperature and or for paleo nutrient concentration. So Neve now has a second very large set of samples to work on. So that was really great. This is just an example of where we collected our data. We tried to collect 3 times a day. And then anytime we saw anything unusual happening, we all carried our phones around with us with Grafana so we could see, oh, we're in a bloom or the temperature has changed. Let's go run and take a sample. So you can see the salinity here on the left, the temperature, so a big triangle. But the last is a blow up of this little area here. And, fortunately for us, Fiorenza was very, very, very attentive. And she noticed that we probably ran into a potential heat wave as we were transiting, the temperatures across. So right in there, and then also it was a lot warmer on the way back than on the way down, and that's we're not sure if it was a heat wave. Are there eddies? Is it just some type of spatial variability in the distribution of water masses? But that's something that we can address hopefully with Izzy and in the future with some physical oceanography. So it was a pretty exciting and easy data set to get, despite spending maybe 5 hours a day filtering.
So I was on the deep-toed camera team, so we were looking at the benthic invertebrates all of the animals on the seafloor. You can see here, this first map shows all of the deep-toed camera deployments that we did across the voyage. So we had 18 deployments, 15 of which were successful transects along the bottom, and in a depth range of about 300 to about 2,200 meters. So we had a really cool sort of range of different habitats that we were able to sample with the deep-toed camera. We had this one right up the top there, which was on that mid-ocean ridge that Luca was talking about before. So that was sort of an opportunistic deployment of the camera that we did there. And then you can see here is a zoomed in map of the sites that we sampled in the Dibble region. So the one right up the top is the one on that refugia hypothesis area. We sampled in the basin as well, and then on the slope and the shelf. So I'll just show some of the sort of highlights and exciting photos that we found along the way. So this sort of area of biodiversity here, you can see sponges and soft corals and crinoids, and there's some fish in there as well. There's some hydrocorals too. More fish hanging out in sponges and crinoids and stuff like that. We found in some of the first sites that we sampled in the Dibble region, a lot of these ice fish with their fish nests. So you can see like these sort of fish nests here. So that was really cool to observe. Anemones and soft corals and sea urchins. We also, so you can see the bathy data here showing the iceberg scour. So this was on the shelf at the Dibble region. So we were able to sort of observe those areas of iceberg scour as well, which was really interesting. You can sort of see here, there's a rhino hanging on the side of where there's been a scour. So we saw quite a bit of that as well. One of my favorite photos of this Umbellula species, which is a type of soft coral, and then the octopus hanging out in the background. Sponges. Occasionally we saw a big dropstone, which often had a lot more biodiversity on it than the surrounding areas, which was really, exciting to come across with the camera. And also we were able to take some photos and videos of the seafloor with the CTD as well. So we had a downward facing camera on the CTD. So of all of the 37 CTD deployments that we did, we were able to take a 4-minute video and some stills along there as well. So that sort of added really greatly to our spatial distribution of our samples. So this is just an example here with the acorn worm, and then this fish, which we saw a lot of in the CTD camera, that was an abyssal grenadier. And that's it for me.
Hi, I'm Sally. So I'm part of the SLED team led by Jan Strungnall, and there's only a team of 3 of us on the ship. So we're really grateful that everyone else on the voyage helped us to collect our animals. So for us, seafloor biology in general from the continental shelf of the East Antarctica is very understudied. So any samples from this region, whether it's from Dibble, Mertz, or the Cook Ice Shelf region, are very important to us. So for our objectives, we wanted to collect genetic samples of marine invertebrates of the continental shelf and also the slope area to understand diversity, connectivity, and biogeographic history of the animals, but also for conservation purposes like the distribution of genetic diversity around Antarctica, use the genetic to understand ice sheet history, and also in general to understand the forces that have shaped the evolution of life in the Southern Ocean, like the refugia hypothesis, like where the animal lived and persist during periods of extreme glaciation in the past. So for us, we deployed a total of 13 sleds, which is very good. 12 sleds were done on the continental shelf and the slope in the Dibble Glacier region, but we also managed to do 1 sled in the very deep water in the Mertz Glacier region and collected a really, really interesting animals from there. And in total, we collected 480 marine vertebrate samples that are very high quality, and they were all preserved for whole genome, reference genome, and population genetic analysis that would certainly last us forever. And I just want to highlight that even though we're biologists and we're geneticists, we really rely on subbottle profiling and the multidisciplinary aspect of the voyage to make sure we target the best site possible for our questions. Here's an example of some subbottom profiling that was done and on the ship, and we rely on this data to understand the topography and the slope and where there is a area of sediment or where there's a rocky area to allow us to decide whether the location is interesting and good enough to deploy our gear. And there's some photos of our process, how we do it. We get the sled, put the sled in the water, get the sled back up, get some kin beans to go into the sled and see where the animals are hiding in there. It's a very muddy process, and once we get all animals, we put them in the lab in a trace of water, seawater, to clean them, preserve them, take individual pictures of them, and identify them to genus and species level. And on this voyage, we collected a lot of animals of our interest, and we mostly ticked off our target species list, which is very good. and collected a lot of octopuses, which is one of our targets taxa, but also perhaps a new species of octopus that we'll do some genetic analysis to verify, and maybe it can be called the Embreti.
Hi, my name's Jolene, and while it's fantastic to be on the expedition and see all the great science being done, my role was to get it out to the general public and also to the schools around the world. So in addition to having a marine background, I was also previously a teacher of marine science, and so I did outreach from the investigator to over 48 sessions. I'm unsure, I think it's about 35 schools at the moment, because a session could include just one class, or it could include many classes from many different schools, and including a session that I did with the Great Barrier Reef Marine Park Authority and the Reef Guardian program, where they had 10 schools plus about 50 homeschoolers involved. So all up, in the sessions, I reached about 2,500 students across the world, both with the scientists back in their home countries, doing some to the university and to their schools, or contacting their previous primary schools and high schools. And then also, of course, in Australia, I'm based in Queensland, so a lot of the Australian ones were primarily there. But we reached a lot of the students in the classroom and aligned the science that we were doing on the ship with that they do in the classroom. So talking about evolution, talking about habitats, talking about the adaptation to Antarctica and things like that. And in addition to that, of course, it was also included all the teachers and the adults that were in the homeschool with them. And so that word will spread, basically. So getting the science out of the journals and to the community, and hopefully involving those students coming up in the future. So that was the breakdown by primary and secondary, and then this is the breakdown by country. So we're able to do 5 different countries, mainly because it was associated with the researchers that were on board, and as I said, to their home schools or universities. And often, like in the pictures there, Lara or Anna and Luca, and so all helped with the translation sometimes, and Linda did some as well. So some of the things that the students and the teachers then send us, I think it was really cool. So before coming on the expedition, I contacted a lot of the Queensland schools, and it was really awesome for me that Moranbah East State School, which, if any of you know Queensland, that's a very small mining town out east of Mackay, and they had their 4s, 5s and 6s join on 3 mornings. And yeah, so it was a cohort of about 80 each time. And then they, so it was really good for me that they, being a mining background, were so interested in the research in the Antarctic. The school in Portugal put up a post. So I also did social media, which I'll talk about in a moment. And then one of the schools that Jan talked to in Germany, all the kids did a poster each, and then the teacher put that together and sent that through to us. And then Linda had brought some animals from, little stuffed animals from a school in Germany. So they did a tour of the ship, helping out all the scientists and the ship's mate driving the ship. And so we sent that poster to the school to show that the animals had been helping us out on the voyage as well. And they were really excited about that. And then social media, and I apologise straight up for Grace that I forgot the blogs in here, but we had outreach on Facebook, Instagram and LinkedIn, and also on the blogs. which were very well read and well received. It's only me getting the information out here, so encouraging the scientists to also extend to their social groups. But I feel that C2 School reached quite a good number of people in the community in getting the science out there, and also the crew. The crew, I also had sessions with some of their kids as well, and they are excited to see what their parents do on the ship as well.
Yeah, so you heard a lot of these numbers already, so I'll just point out a few things on this list. The total distance we traveled was over 6,000 nautical miles, and over 4,000 nautical miles of that is newly mapped. So that was quite an achievement. We have some numbers here on how far we got south, east, and west. I think one to point out was the biggest roll. That was 21.4 degrees on the way back, which felt quite intense, with chairs, with us sitting on the chairs and the chairs sliding across the room. You've heard a lot of these numbers already about how many samples we took. It'll keep us busy for the next decade, probably. Everything in pink is way beyond what we thought we would collect. So that was great. We also collected a record piston core. This was the first 5-barrel piston core that was collected on the Investigator, and that meant about 24 meters. So we recovered 20.5 meters of sediments, which was a really good recovery. You can see a number of photos from that day. I'd say, along with the day that my baby horse let me sit on her back for the first time, that was probably one of the greatest days of experience. I even took a photo of the time. So this was the 17th of January at 8:17 when the piston core hit the seafloor. And yeah, it created a lot of anxiety coming up, but it was a really good day of us just out there and cutting the core and taking the samples. We also actually, I think we might have also broken a record with bending a piston core as much as you can bend it. Took a whole day to bring it back on the ship. So I didn't put any photos of that, but I can show you if you like to see it. All of that, of course, wouldn't have been possible without this absolutely amazing team. It's been such a great experience. I thank every single one of you for being there and doing all your hard work, being so enthusiastic about it, and just sharing your knowledge. It was a really, really good team. I'd also really like to thank the Marine National Facility, the crew, all the technical support we had, Frank Nietzsche, who helped us getting our head around the Dibble Glacier, and then, of course, SAFE and ACS, who supported a lot of the early career researchers that otherwise wouldn't have been able to participate in the voyage. So thank you very much for all this support. And I think I'll leave it there. Yes.
Yeah. It's not because I forgot, but I also want to thank IMAS and CSIRO, because not just the scientists were gorgeous, but also the technicians and the crew. We were really a very good team, and I was very impressed about how the efficient machines travel system. So again, thank you all.
So we'll be around if you have questions or want to chat. We have time.